Thank you for the invitation to speak, and I've really enjoyed seeing some familiar faces that I've not seen in a while. I was going to take just a few quick minutes to share with you a really cool story from my lab that we're hoping to submit the paper on this structure in the next couple of weeks. And it's on a novel immunity repressor that we have a structure of bound to DNA. And this is a really cool story where the crystal structure and the SAXS data agree with each other and each tell something very important about this. So I want to begin. I don't know how much background people have on what bacteriophages are. And so I wanted to start by just with a little bit of biology. And what are bacteriophages? They are viruses that only infect bacteria. And something you may or may not know is that they are the most abundant biological entity on the planet. And so it's estimated that there are 10 to the 31 bacteriophages on the Earth. And they are extremely diverse and selective for their bacterial host, and they outnumber their host bacteria at a ratio of 10 to 1. So anywhere on the Earth you can find bacteria, you will find lots of phages. Now, a couple of things that you may find interesting is first, bacteriophages are recently shown some promise in being used to treat antibiotic-resistant bacterial infections. The reason this matters is it's estimated by 2050, there will be approximately 10 million deaths per year in the world due to antibiotic-resistant bacteria. So this is going to be, it already is a problem and it's only getting worse. What I find extremely interesting is this bottom point is that when you look at phage proteins, we have no idea what approximately 80% of the genes do. Okay. And so as a protein biochemist, I find this extremely fascinating. And I think my idea is that if we look at the structures of these proteins, we're going to find some novel protein folds. One thing I wanted to point out is when you think about viruses and how viruses work, you probably think of what's going on this side of the slide, where a virus will find its host cell. In this case, it's a poor little bacteria. And the virus will come in, it will inject its DNA, and then that cell will be hijacked to become a virus-producing factory and ultimately kill the cell. Well, that's not what always happens. And if you look on the right side, we have what's called the lysogenic cycle, and where the virus will inject its DNA, and instead of killing the bacteria, it integrates its DNA into the host chromosome. And then the viral DNA gets replicated with the cell as the cell divides, so on and so on. And so on the right side here, the virus can lie dormant until it needs to leave to make more virions. And so I bring this up because if you want to produce a virus that can kill antibiotic-resistant bacteria, you're going to want the lytic guys on the left. You're not going to want these what are called temperate phages that do the lysogenic cycle on the right. And so there's a protein, a really important protein that drives the lysogenic cycle, and it's called a repressor. And a repressor is simply a protein that's present in temperate bacteriophages. And what they do is they bind to a target DNA sequence to prevent lytic gene expression so that they can stay in that lysogenic cycle. The most well-studied repressor is from phage lambda that infects E. coli, but I want to talk about a phage known as L5. It is a mycobacteriophage, which means it infects mycobacterial species. And it's a protein that's been well characterized, a variety of papers, primarily from Graham Haffel's lab. And the L5 repressor binds to this specific DNA sequence here. And what's really neat is that if you look at the genome map of the phage, all of these circles show the binding sites for this repressor. So essentially what the repressor does is it coats the whole genome to prevent the transcription of lytic genes. Now, if we think about this L5 mycobacteriophage repressor, what my lab became really interested in is there's not a structure of this protein. And if you look at the primary sequence, which is shown up here on top, the blue region is a predicted helix-turn-helix DNA binding domain, and that makes perfect sense based on what this does. But all of the amino acids shown in black, we have absolutely no idea what the function of those is or what the structure of those is. And this goes back to this idea of novel protein folds. The second thing that to point out is that the L5 works as a monomer, and this is quite distinct from other repressors such as lambda, which functions as a dimer. And so if this thing is indeed a monomer, the question we had is how does it perform the same function that typically is performed by proteins that form dimers and even higher ordered oligomers? So a few years ago, I reached out to Mikkel and Daniel to get some SAXS data on this protein. We did not have the structure at the time. And all I wanted to do was I had some DNAs of different sizes, and I have highlighted in red, this is that sequence that this repressor preferentially recognizes. And what we were doing was we were simply making the DNA longer to add flanking sequences on each side of the recognition sequence. And we wanted to see what this protein could bind to answer two questions. One, is it indeed a monomer on DNA? And two, what size DNA do we need for this protein to bind, with the idea of trying to get the structure of it? And so this is showing the SAXS data that we collected on all of these samples. They're color coded, and you can see the RFG and Dmax values for these guys. And one of the things that I noticed was that when we went to the smallest DNA, just this 13-mer that just has the recognition sequence, I noticed that the sample is getting quite small in terms of its Dmax and its radius of gyration. Now, if we focus on this 20-mer sample, what we saw when we did SEC SAXS mols was that we did in fact confirm that this protein was a monomer on DNA. Okay, so looking at this sample here. So that was the first thing we wanted to address. But if we compared this DNA sequence to this DNA sequence here, the other thing that we noticed was that the protein was not able to bind to the smallest DNA in our samples. And so this green trace here, sorry about the confusion on the colors, but this green trace here represents the 20-mer duplex DNA. And you can see that we formed a nice protein DNA complex when we look at the elution times compared to the free protein shown in dark blue. And then the light blue represents the smallest DNA sequence. And so based on the SAXS data, we then went to the to set up crystal trays with the mindset that this DNA is too small, and this DNA seems to be quite large enough to get crystals. And so long story short, we were able to get crystals with a 20-mer. We were not able to get crystals with the 24, which told us that probably the DNA was too long and was kind of flapping in solution. And our optimal crystals came when we increased the DNA size to a 21-mer. So fast forward a couple of years, because this protein was quite challenging to get the structure for a variety of reasons that I won't go into here, but we were able to get the crystal structure of this protein to 3.15 angstroms resolution. And it was done on the 21-mer DNA. And this protein is indeed a monomer on the DNA substrate. And I want to point out, this is that helix turn helix that was correctly predicted from the sequence. And then this orange domain, which is we're really excited about, is a second novel DNA binding domain. So it was not predicted that this part would bind DNA based on the sequence alone. And when we compare this to other structures in the DALI, using the DALI server, we do not see any structural hits representing that this is a novel fold. And the other neat thing is that this map up here shows all the residues that are binding to the DNA. And our SAXS data told us that just the recognition sequence, which is underlined, was not big enough. And what we see is that we have two residues over here and one residue over here that is binding to DNA just outside of the consensus sequence. And this tryptophan 50 is absolutely essential for the function of this protein. And so that was something that was really exciting for us to see. The SAXS data told us this, and we can now explain that in the crystal structure, especially related to this tryptophan 50. I just want to show the same structure in a surface representation. It's still color-coded the same. We call this little middle region here the helix bridge, because it sits above the minor groove, and we have two DNA binding domains binding at adjacent major grooves, very similar to what we see in phage lambda. And so the final thing that I wanted to show you was, after I got the structure, I went back to Mikkel and I said, now I have the structure, can we go back to the SAXS data and just do some comparisons to see how the structure compares to the SAXS data? And long story short, it agrees beautifully. This is my crystal structure shown up here. And what you see is the green trace shows the fit of this structure to the SAXS data with a chi-square of 1.75. It's kind of hard to see, but there's some light blue regions shown in the structure. Those were disordered in the crystal structure, so we had to build that in and do some rigid body modeling using BibleMD. And so this was the 20-mer duplex data shown here with the crystal structure in green, a beautiful fit. And then finally, you remember I said it looked like the 13-mer was too small. When we compare the SAXS data to our structure, what we see is that the best fit comes from if we have a mixture of the free DNA and the free protein in solution. 64% of that being DNA, 36% of a protein with a chi-square of 1.2. When we try to fit this data to the crystal structure, we get a really poor fit. And we also get a poor fit when we fit it to just free DNA. And so what's next for this? Like I said, we're wrapping this up. We're hoping to submit the paper in a few weeks. Where we're at right now is Mikkel and I are talking about just the protein only data. I didn't really talk about this in this talk. And the reason is that the protein seems to be very unhappy off of DNA. And so it's been a bit challenging to analyze the protein only data. And so Mikkel and I are working on that now, but it looks like that it is also a monomer, and we're going to be able to do some modeling of that as well. And then we have several additional proteins like this currently in our research pipeline. A lot of these repressor proteins recognize different DNA sequences. And so some of my future goals is to pump more of these through the SAXS pipeline to learn more about their oligomeric state, DNA binding preferences, and also conformational dynamics, both on and off DNA. And so with that, I just want to thank everybody that's a part of this project. But for the sake of this talk, I really want to thank the ALS. On the crystallography side, I use Beamline 831, and then I want to thank Mikkel and Daniel at SIBYLS for their help with the data collection and the processing. And with that, I'd be happy to take any questions.